This is lecture 1 in chapter 3. Today we're going to talk about the cell theory and then the plasma membrane and how substances move in and out of cells. So we talked early in chapter 1 and 2 about cells being the basic unit of life. And so there is a thing called a cell theory, and there's 3 parts to it. It is cells, the basic unit of life. All living things are made of cells, and new cells come from pre-existing cells. So cells arrive from other cells. So when we talk about how cells are organized, we talk about eukaryotic cells. There are prokaryotic cells, they don't have a nucleus, but because this class is primarily about body systems, we're going to talk mainly about the eukaryotic cell. And so the eukaryotic cell is a bunch of little organelles in it, and it can live by itself. That's like a eukaryotic bacteria is just a little one-celled organism. It can cause lots of havoc in our bodies and also help us a lot. We have a lot of bacteria that live in and on us, and it helps us be functioning. So when we'll talk about cell parts later, but today we're going to primarily talk about the plasma membrane and how things move in and out of cells. So your plasma membrane is made up of a phospholipid bilayer. Phospholipids are members of the lipid family, and we talked about lipids last chapter. They repel water, and they are hydrophobic, which is a good thing. That's why cells are the basic unit of life. Essentially, the plasma membrane does not dissolve in water, and it's kind of like the cell skin, and it allows things in and out. It can allow the small substances in and out to diffuse through the cell membrane. Then it needs channels and gates for other things, and then sometimes it just engulfs really large particles to eat. And we'll talk on about that as we move forward. So here's a cute little picture of the plasma membrane. If we were actually in a class, if you were in a biology class of mine, we would illustrate this and draw it out and label everything. But as it is online, we just take our little online class and move along. So this is a picture of it. And when you look in here, you've got these right here are your phospholipids, and they are nonpolar. Okay, so they have this bilayer, and that's your protective layer. Embedded in that bilayer are proteins. All different kinds of proteins. There's proteins that tell cells what their names are. There's communication proteins. There's channel proteins, which allow bigger substances in and out. There's cholesterol in here that allows the plasma membrane to have some shape. And your PowerPoint will go into greater detail about what's in the plasma membrane. But right now I want to kind of chat with you. That's kind of what our lectures are. It's not a real lecture. I'm just kind of chatting about your talking points of these chapters. So the channels will let some things in and out, and tiny little hydrophobic substance can just pass freely right through that plasma membrane, things like carbon dioxide. But you've got your ions, your large particles and molecules, they need help getting through there. Water can cross the membrane, but they do that through channels called aquaporins. So there's some different ways that substances cross the plasma membrane. Diffusion and osmosis. Then you've got facilitated diffusion. All 3 of these guys are passive. They don't take any energy. And then you have active transport, endocytosis, and exocytosis. These do need energy to work. So these are active processes. So when we're talking about diffusion is just the movement of particles from areas of high to areas of low. So you think about concentration. Like if you put some food coloring in water, that food coloring is highly concentrated. If you leave it, it'll eventually disperse, okay? And these molecules are going to move until they are equally distributed. This happens with scents. Like if you spray some perfume in the air, all those particles are close, they eventually move out. Now, osmosis is just the movement of water, okay? And this is from areas of high to low, ready? And so when we're thinking about that, because we're really talking about cells and we're talking about living things, our blood cells, okay, are isotonic to their environment. The inside and the outside are isotonic. And that is what your normal body fluids are. When you have an IV, it's got 0.9% salt solution in it. Your blood cells are 0.9% salt solution. And so the water moves in and out from your blood cells, leaving them nice and puffy and full of hemoglobin, and we don't die. It's a beautiful thing, right? Now, when we talk about the other things, though, we're looking at the solute concentration in the environment of the cell. And so hypotonic solutions, the solute concentration in the environment is lower. Okay, it's lower in the environment than it is in the cell. And so the water is going to move into the cell, and the cell is going to puff up and eventually explode. Okay, hypertonic means that the solute concentration in the environment is higher. And so what's going to happen here is water is going to move out of the cell, and the cell is going to shrivel up. Okay, and osmotic pressure is what drives this. So when we look at our little blood cells and we look at our little picture here, okay, the isotonic solution leaves those blood cells perfectly. They're kind of like donut shaped. They've got that little concave piece in the middle. Hemoglobin is deep in there to carry those oxygen to the cells. And it carries oxygen all through our bodies. So hypotonic, you see the cell kind of puffing up. It'll eventually burst. And then the hypertonic solution, it shrivels up. Now, diffusion and osmosis are both passive, require no energy. And neither does facilitated diffusion. Okay? Facilitated diffusion has help, because that's what facilitated means, is help. So you've got these little protein channels, and they move things in and out of the cell. No energy required. So if you look at our little picture here, see this goes just in and out, right? Glucose, really easy, but it has to have that channel. If there wasn't that channel, glucose would be too big. Remember, glucose is a carb, and it would be too big and it wouldn't be able to get in. Now we talk about active transport. Active transport is the movement of molecules from a lower to a higher concentration. So that actually uses energy, and it also requires a protein carrier. And we usually call those protein carriers a pump. So we think about using a pump. If we are using a gas pump, right, to fill up our car, we're squeezing it and using energy. If we're filling up a bike, using a bike pump to fill up the air, you're pushing it down and filling up that bike tire, right? Using energy. We have to use energy in active transport. And we're moving stuff from lower to higher. So it's kind of like, I like to think about diffusion is kind of like high to low. So you're like pushing me down a slide. Doesn't take any energy, you just push and I just go. But if you have to push me up a slide, it's going to take you a lot of energy because I ain't a little girl, right? So when you think moving from higher to lower versus lower to higher takes energy. Okay, so sodium potassium pump's your most common pump in a cell. So then when we think about bulk transport, bulk meaning big, we're going to talk about exocytosis and endocytosis. Okay, so your examples of bulk transport is exocytosis and endocytosis. Okay, endocytosis is just taking in particles. Okay, there's two different types. There's phagocytosis, taking in a big food particle, or pinocytosis, taking in liquids. Exocytosis is excreting large particles. Okay. Exocytosis is just like reverse endocytosis. My kids in high school used to say it's like cell poop because it's just expelling the large particles. All right. So that is section 1. You will get another section of cell parts with this chapter. You will not get a lecture for chapter 4. It's pretty self-explanatory. And then we'll talk more about DNA and RNA next time. Remember, if you have questions, email me. I'm available and I can help. Have a good day.